Cell signaling is crucial in a changing environment as it enables cells to communicate by receiving and sending out signals. A ligand is any molecule that binds to something else to perform a biological process. A hormone is a ligand that acts as a signaling molecule. Hormones are part of endocrine signaling. Cells have proteins called receptors that are unique to the signaling molecules that bind to that receptor. In the case of adrenaline, there is an adrenergic receptor. When the adrenergic receptor is activated, it triggers the G protein coupled receptor. When the G protein is in its inactive state, GDP is in its binding site. Once the adrenaline binds, an allosteric change is triggered and GDP leaves, being replaced by GTP. The activated G protein bumps into adenyl cyclase, causing a shape change. Then a phosphate is released from GTP, inactivating the G protein, leaving GDP in the binding site. Adenyl cyclase is an enzyme that catalyzes the conversion of ATP into CAMP, a second messenger. Phosphates are removed from ATP to form CAMP. CAMP will amplify the response in the cell by activating multiple proteins. The CAMP molecules activate the enzyme protein kinase A, which phosphorylates many protein substrates or relay proteins by attaching phosphates to them. The phosphorylation cascade can be regulated by turning off the signal. Phosphatases are able to catalyze the hydrolysis of phosphates that were attached to the proteins in the cascade via protein kinase A. This phosphorylation-dephosphorylation system is a switch in the cell that is able to turn activities on or off depending on what the cell needs to do. After the processes of reception and transduction, finally the signal triggers a cellular response. In this sense, we will be looking at how these signals turn on and off genes. The first process is making mRNA, also known as transcription. Then, specific mRNA are made into a double helix and are attracted to move toward a ribosome. After all, finishing the process of translation.